We know these treatments are clinically effective based on multiple randomized clinical trials. We also have these very interesting novel treatment patterns, accelerated patterns where you can apply multiple treatments in a given day. The new SAINT protocol by my colleague Nolan Williams was recently FDA approved, I think about a month ago now, to put 10 treatments in a single day. So the treatment only lasts a week. So lots of excitement and innovation in this space, but we still lack a mechanistic understanding of what TMS does to the brain. We don't have brain markers to stratify, predict, and track, and we can't selectively modulate this biomarker because we don't yet have it. So what do we know? We know that single pulses of TMS, the capacitor gets discharged, so time-varying magnetic field. That field gets into the skull, induces an electrical current, and activates neurons. We know this from single pulse studies in non-human primate visual system, can induce action potentials within 10 milliseconds, where sham stimulation, a flipped coil, does not do this. And we know in humans, single pulse TMS to the motor cortex induces a motor evoked potential through the corticospinal tract. No one disputes this. But the question we have is, how does repetitive TMS induce brain plasticity? So this work all comes from animal slice physiology in the 70s, where high frequency electrical stimulation, usually 100 hertz to the hippocampus, can induce long-term potentiation. Try to bring that up to humans, and you can't use electrical stimulation because it won't get through the skull. So you turn to TMS. You can't use 100 hertz because you would induce seizures. So you bring that down to 10 hertz, and you're left with something that gives us something like this. So 10 hertz patterned TMS to the motor cortex. When measuring the motor evoked potential change, something that has very high signal to noise, you don't see a clear effect. Similarly, these newer patterns, continuous theta burst, which is thought to be inhibitory, and intermittent theta burst, which is thought to be excitatory, across a large number of subjects showed no net effect. Outside of the motor cortex, where we don't have this high signal to noise measurement, it's even more confusing. So what do we think we're doing? Well, we think in depression, we have a hypoactive dorsolateral prefrontal cortex, a hypoconnected frontoparietal network. We have less top-down inhibition to the default mode and salience network, and those networks are allowed to run awry and underlie a lot of the symptoms that we see in depression. We think TMS can upregulate or induce long-term potentiation in the dIPFC, normalize the frontoparietal network, and normalize those other networks. There's a little bit of imaging evidence to suggest this, but it's not super clear. So as a physician engineer, I would love for my patient to walk into my room, and I would be able to do two things. One, figure out what brain circuit is abnormal in their brain. And two, have the tools in our toolbox to be able to enhance or weaken that circuit. If we could do that with TMS, we'd have a treatment with unlimited applications with minimal side effects. TMS is FDA cleared for the disorders in bold, and my guess is by 2030, all of these on the list will be FDA cleared, which is exciting because it represents an enormous potential, but a little scary that we know so little about what we're doing. A hidden agenda here is for basic human neuroscience. We don't have the ability to temporarily induce LTP or LTP and measure the functional and behavioral effect on that circuit, which would be hugely valuable. Put another way, we need to learn the language of TMS. So we know the sound, we know what a single pulse does. My child is learning to speak, and he says two words. He says mine, which means mine, and bablos, 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 which means bubbles. I think this is where we're at with TMS. We have two patterns that we say over and over and over again. We receive no feedback, no context. We don't try to piece words together. We're missing everything else, right? We're missing all of the other words in the language. Context, and there's a lot of context. We need to start with a single word or a single stimulation train, figure out how to quantify it, and then we can explore how other words push that biomarker around, and then we can piece them together. We can explore this multidimensional parameter space. We can start personalizing by picking peaks in this multidimensional space that maximally modulate that biomarker for that individual, and we can adapt treatment. It all depends on this biomarker, right? Or set of biomarkers. So why aren't we there? Like I said before, we don't have that objective biomarker that either tracks a circuit excitability or depression. If we had that, we could track it in real time. We would have a metric of target engagement that predicts symptom change. I feel strongly that without this marker, we can go and try to innovate in the treatment space, but a lot of what we'll be doing is trial and error guesswork, and we'll be utilizing a huge number of resources to do a large number of clinical trials. And we want to reduce those dimensions and bring it down to something where we can tangibly measure the biomarker on a millisecond basis and explore different treatment patterns. This multidimensional parameter space is a little scary, right? Location, frequency, pattern, amplitude, duration, state dependence, number of sessions, intersession spacing. I really could go on here. So we need to start thinking about this multidimensional space, especially as we get to these accelerated treatments where we're putting 10 in a day with some spacing that may or may not be maximally effective. Resources I mentioned before, to do a clinical trial of 10 hertz versus theta bursts takes hundreds of patients, 30 treatments per patient. It's thousands of days of TMS, which will overwhelm any small lab, right? So in our lab, we bring that down to a single day and look at single doses of TMS. And in terms of number of subjects, we bring it down to a single subject and try to work to enhance the signal to noise that we can see statistical difference on a single day, and do crossover randomized design studies. And finally, with this marker, we want to be able to monitor and adapt in real time. Okay, so this is our lab, founded during COVID. We focus on neuromodulation and human electrophysiology. We deconstruct brain stimulation to establish the fundamental mechanisms underlying human neuroplasticity and try to develop real-time closed-loop treatments for mental health disorders. We do this 3 different ways. We develop novel methods to probe the human brain in a causal and directional manner. We try to better understand the mechanisms underlying human brain plasticity and trying to create these real-time closed-loop solutions. We have 3 different preparations that we typically work from. One is a fully invasive approach. So these are medication-resistant patients undergoing surgical evaluation for epilepsy, have up to 300 electrodes placed in their brain to monitor their seizures. During this time, we can focally electrically stimulate, let's say the dorsolateral prefrontal cortex, with a pattern of stimulation that we use clinically for TMS, and just ask with no perceptual change, with very high signal to noise in terms of measuring near the neurons, what changes, why does it change, how long lasting is that change? But this is in epilepsy and it's with electrical stimulation, right? So on the other side of the translational arc, you have non-invasive TMS paired with EEG. You can get this into the clinic. You can do this in healthy controls in patients with depression. You have a clinically utilized tool with TMS, which provides less focal stimulation, EEG recordings that are further away from the neuron, and a lot of stimulation artifact and off-target sensory effects. So you have to deal with this noisy environment, but is clinically very translational. And then we have this new kid on this block, this TMS IEEG, which is sort of a mix of the two, but applying TMS and recording with high signal to noise inside the brain. So I'll introduce that. My hope is that we can bounce ideas back and forth between here. We can get the sort of ground truth approach from the leftmost intracranial electrical stimulation, and then see if we see those signals non-invasively. We can do a clinical trial with TMS EEG and then explore the underlying mechanism invasively. And you can take this one step further if you wanted to. And the 4th leftmost would be animal electrophysiology with single neurons. So a near-term goal here is to develop objective brain biomarkers that enhance their reliability and validity. When I say reliability, I typically refer to internal reliability or test-retest reliability. Typically utilizes the fact that you need to increase the signal to noise via online and offline experimentation to increase your confidence in the signals. And validity, I usually think as biological validity. We need to combine invasive and non-invasive measurements to biologically ground these signals. So I'm going to start introducing the intracranial approach relatively quickly and then focus most of the talk on the non-invasive work. So intracranial EEG, I think I introduced it before. But we have these grids, strips, and now often depth electrodes that are implanted into the brain. We can localize where these electrodes are, and then we can stimulate. So I helped way back when co-develop cortical evoked potential, or CCEP mapping, which is very simply applying focal electrical stimulation to bipolar adjacent contexts and measuring the evoked potential. 10 to 15 milliseconds, you see a deflection in distant regions. Direct, causal, directional, which is pretty cool. We can use this tool in a lot of different ways. One is explore seizure networks. You can look at functional brain networks, do these CCP maps, map out the default mode network. You can use these tools to validate non-invasive recording methods like functional MRI. You can also use this metric to measure brain plasticity inside the brain. So this is one patient. This is their grid and strip distribution. We applied focal electrical stimulation to the prefrontal cortex and measured in this parietal cortex, as well as everywhere else, but this is just an example. This is what the CCP map looks like. A single trial, you can see the negative deflection and stimulation artifact. You can see a strong evoked response on a single trial level, which is pretty nice. When you plot this on the brain, you can see that single pulses of electrical stimulation activates or induces an evoked response locally and also in this parietal region, probably mapping out the frontoparietal network. Then we apply our repetitive electrical stimulation, patterned just like TMS. So 3,000 pulses, 5 seconds on, 10 seconds off to the dIPFC. And using these CCPs after stimulation, just ask what brain regions change, how long are those changes, can we predict those changes? This is just one example of this parietal site. And this is what we saw. So we see a reduction in the early 20 to 40 millisecond evoked response that lasts for about 20 minutes. We were able to predict which regions would undergo these changes based on, as you would guess, the underlying anatomical, so how close to the stimulation site are you, and functional connectivity, how strong were the baseline CCEPs. We went one step further here and we dove into the actual stimulation and could quantify the evoked response to each pulse within each train and just asked, what does a single train of 10 hertz do? How do repeated 10 hertz trains build over time? And we could sort of fill in this space of the induction phase of plasticity. So it raised more questions than answers, as a lot of studies do. But the main ones were, can we do this non-invasively? Can we predict which regions will undergo plasticity before we apply TMS? And can we use this to adapt treatment? I'm just going to introduce the TMS IEEG, which is really brand new. This is a collaboration that we have that's ongoing with the University of Iowa. This is Hiroki Oya in the neurosurgery department, and Aaron Boas, who's a neurologist who does TMS as well. Hiroki did a lot of the phantom testing, so put electrodes inside the phantom and applied TMS. And long story short here, it showed that TMS, single pulses or repetitive TMS, does not induce heating in the electrodes, doesn't displace the electrodes, and the voltage gradients that you see at the electrodes are that predicted by the drop-off from distance from TMS itself. There's no eddy currents or secondary currents. Aaron then did clinical testing and is implanted with 20 patients that have implanted electrodes, applied TMS, single pulse TMS, repetitive TMS, single trains of TMS to multiple different regions, and have shown no adverse events outside of what is typically assumed with TMS, a slight headache, some scalp discomfort. And then we were given the opportunity in 9 patients that got single pulse TMS to the dIPFC to measure the evoked potentials in the brain. So the two conditions are active single pulse TMS every 2 seconds compared to a flipped coil, which is sham stimulation. This is what an intracranial evoked potential looks like. Single electrode, single patient. The single pulse comes at the stimulus artifact in gray. And you see the strong evoked response with TMS compared to sham. We saw some electrodes where you don't see a difference, but you see a strong evoked response. Presumably this is sensory regions. The auditory cortex lights up from the auditory click from TMS. And then other regions showed no effect. When you plot this on a brain on an N of 1, you get something like this. You see regions that show strong evoked responses from TMS and not from sham. And then you see other regions like bilateral auditory cortices where you have the off-target sensory effects. So on a group level, we asked two simple questions so far. A, does TMS induce local effects at the dIPFC? To answer that, we have a slightly complicated figure, and I need to change this. What I want you to get from this image, this is a group response for 9 subjects. Green is where the TMS stimulation site is, neuronavigated, and you see all the electrodes within 3 centimeters across these 9 patients. So there were some electrodes that did respond, that showed strong evoked potentials compared to sham, and a lot of electrodes did not respond in white. And actually only 18% of electrodes in the local dIPFC within 3 centimeters showed a strong significant response, which was somewhat not predicted. The first question we asked is this related to the electrical field distribution? Is it that if the electrical field is maximal at a certain site, you're more likely to find an evoked response? And that answer is kind of. So in regions that had a strong electrical field, again, this is all local, it was more likely to show an evoked response, but this correlation in the scatter plot is like 0.44. So it doesn't explain much of that variance. It actually aligns with some of the research coming out in a parallel field of TMS fMRI, showing that they don't see a bold response locally under the TMS coil. There's a lot of reasons that could explain it, and we're sort of starting or trying to figure that out. What was interesting is when we look at downstream regions. So if we just look at the whole brain, dIPFC doesn't light up the most. That there are other regions, and that is the anterior cingulate cortex and insula, both ipsilaterally and contralaterally, just light up. You can see this on the group brain plot here. These are 3 patients that showed strong evoked responses in the ACC after single pulse TMS to the dIPFC. The first questions that arose here is this due to the saliency of TMS? Is it due to the somatosensory feeling or the pain? All of these networks are sort of in the saliency network, right? So we did a number of control analyses that we're still working on. Step 1 was in these 9 subjects, we looked at the resting functional connectivity between our stim site, and we showed strong functional connections in this ACC region. Step 2, which we only had 2 patients with so far, but 2 patients had both TMS and single pulse electrical stimulation to the dIPFC, which has no perceptual effects. And we saw strong evoked responses in the ACC. And step 3 was we applied TMS to the parietal cortex and didn't see this ACC activation. So we're still sorting through this, but it's certainly interesting. Summary here on the intracranial work. I hope to have shown you that the electrical stimulation can be a powerful tool to try to assess pattern stimulation in a sort of ground truth space with no perceptual effects. That 10 hertz electrical stimulation induced brain changes for 20 minutes in a predictable manner. And that this TMS intracranial approach, again, brand new, a lot of this work is preliminary, but it's safe. It induces local responses, but a very small number of them, not fully explained by the electrical field distribution. It reliably evokes these downstream effects in the ACC and insula, that the dIPFC was functionally connected to these regions, and the parietal cortex did not show this response. So there's a lot more to do here in this TMS IEEG space. We just got research grade TMS compatible amplifiers, which we did not have before. So hopefully we can resolve a lot of the signal in the early first 30 milliseconds or so that we were not able to see in this study. We're looking at dose response curves, trying to dissect sham a little bit more from the cutaneous and auditory aspects of the sham response, and then starting to look at single trains and even how repetitive trains build up in the brain. So I'm going to shift non-invasive now and sort of talk about where we're at in the field and then where we're going. So currently in the clinic, when we treat a patient, we are targeting the dIPFC using scalp-based methods. We are dosing based on a percent of the motor threshold, and we're treating in an open loop way for 30 treatments daily, typically. Research in this space, there's a hot area of research which involves in targeting, right? So can we hit a spot in the dIPFC that is either functionally correlated or functionally negatively correlated with areas implicated in depression? Dosing, there's starting to be discussions about, should we dose in a different way instead of just multiplying 1.2 times the motor threshold, which doesn't make that much sense. Can we dose based on the electrical field distribution in certain computational models? And treatment, as I said before, accelerated treatment, theta burst, and then accelerated theta burst is the newest thing. Can we put up to 10 treatments in a day, which would allow the whole TMS treatment to be a week, which would be great. And it's already FDA cleared at this point. Our approach is slightly different. We believe in the targeting approach, but we don't use fMRI to do so. We think we can use EEG in the clinic to do so. We dose, but we think about dosing, like I said before, in this multidimensional parameter space. And in terms of treatment, we want to move towards this closed loop application, which I'll explain in a second. So right now we are open loop, one size fits all, hoping to move towards starting with a certain pattern, measuring the EEG response either during a single dose or between doses of TMS, and updating that stimulation pattern, right? It's based on one's physiology or plasticity, presumably faster and much more effective. But to get there, it's kind of a long road. So similar to, developing glucose monitoring for diabetes or blood pressure for hypertension, we want something like that for mental health, right? And we don't have that right now. We need to discover the biomarker. We need to characterize it. We need to optimize it, and then we can apply it. So I'm going to start to walk you through one of these approaches. So we use TMS EEG, like I said before. It allows the causal perturbation of a specific brain region, relatively specific, and millisecond temporal readout of the whole brain, or at least of the whole cerebral cortex. It's relatively cheap, and it can get into the clinic. It's a whole bunch of stimulation artifacts, which I'm not going to dive into, but we and others have developed pre-processing algorithms to clean up the data. I don't want you to read too much into this, but this is what a TMS evoked potential looks like. The dotted line is at time 0 when you apply the single pulse. I just want you to get that it is a multiphasic response that lasts about 300 milliseconds. If you source localize this and plot this on the brain, you get these pretty maps of causal brain dynamics, all non-invasively. To my knowledge, it's really the only tool that can do this in non-invasive human neuroscience. So we use this tool, and we want to find a biomarker with high signal to noise that measures network excitability. We can quantify on a millisecond basis and changes with TMS treatment. We think we have one of these. We call this the P30. Single pulse TMS. The first deflection is a positive deflection, around 25 to 40 milliseconds. It's lateralized. It is the earliest peak in TMS EEG. It's thought to reflect local excitability. It gets suppressed when you add in voltage-gated sodium channel blockers. And we can see this intracranially. I showed this plot before, but single pulse electrical stim with no perception, you see this evoked response. Presumably there might be an earlier initial response, but there's a lot of artifact very early on. So this is the first time we can sort of look at the response. So 3 lines of evidence to suggest that this might be a valuable metric. So we completed in Amit Etkin's lab a few years back. We completed a randomized control study where we applied 4 weeks of active TMS versus sham TMS to patients with major depressive disorder and measured with single pulse TMS EEG before and after that month. We looked at all of the different peaks and troughs of the TMS EEG, and only the early one showed statistical differences. What we saw was a group by time effect. Group is active versus sham treatment. Time is pre versus post those 4 weeks of treatment. In the left prefrontal and parietal cortex, this was driven by the active arm. The P30 actually goes down in active treatment and doesn't change much with sham. The P30 is localized to the medial and lateral prefrontal cortices. And although take it with a grain of salt in a small sample size, it's suggestive that the degree of P30 suppression might be linked to clinical outcome, to clinical improvement. The second line here is we did a study with a single dose of 10 Hertz rTMS to healthy controls and applied single pulse TMS to the dIPFC. And here we're seeing a suppression. This is in the parietal cortex here, but see suppression in this early response after 10 Hertz TMS. The third piece of evidence is the intracranial work, that the 10 Hertz electrical stimulation suppresses that early 20 to 40 millisecond response. So there might be something here. The issue with this signal is it has really low signal to noise, even compared to the other time windows in the TMS EEG. It's a tiny signal. So we need to boost that signal, right? Two different ways to boost it: reduce the noise or boost the signal. We chose to reduce the noise initially. So this was the first study that we did in the lab. There's well-known off-target sensory responses when you apply single pulses of TMS. Hurts a little bit, you have an auditory click, and you feel it on your scalp. And all those come to a strong evoked response in the brain, typically around 100 to 200 milliseconds, but can be as early as 30 milliseconds is when it starts. So you can see how it can confound what we're looking for, which is not this. So we used healthy controls. Our goal is to minimize this signal. The standard way to perform TMS EEG up to this point is to use the TMS click, generate some white noise, and put it in earbuds so it's always playing, and then have some foam between the TMS coil and the scalp, so you reduce bone conduction. We added two other things here. We added, very simply, these construction worker earmuffs to dampen the signal coming in before that white noise, which would be in your ear. And we had predictable interstimulus interval timing. So typically people jitter the single pulse so as not to induce plasticity. But doing that increases the saliency of the signal too, which will increase this N100 and P200. So we actually used a predictable timing, showed that it does not induce plasticity when you just apply 100 or 150 single pulses. Called this attenuate, which is a ridiculous acronym, but kind of fun. This is our main finding on our group level. So what you can see here is in black is with no masking. The standard masking is in blue, and then our attenuate protocol is in red. So we're able to reduce this later time period signal down pretty low. Our signals of interest are left of that in the 30 to 50 millisecond time period, which did not change. So we're almost getting to the level, the signal is almost as high as the noise, but we can do better than that. We also reduce the perceptual effects as well. So in terms of boosting the signal, we actually we don't know that much about this P30 in the prefrontal cortex. So we just wanted to characterize, right? We want to understand how does location and angle affect this P30. So this is a simple offline study, single pulse TMS to these 6 brain regions. We tried to characterize the anterior to posterior and superior to inferior direction, as well as pick targets that are clinically utilized. So like T1 is the 5 centimeter rule, where people move 5 centimeters anterior of the motor cortex to hit the dIPFC. T6 was 5.5 centimeters. T4 is that negative correlation between the dIPFC and the subgenual cortex. So our goal here is early TEP, or this P30 TEP optimization. Let's see which of these sites and angles give the highest signal. And we thought that it would be sensitive to both location and angle and related to muscle artifact. So we talked about the later artifacts that we see in TMS EEG, the sensory evoked potentials. There are much earlier artifacts. There's a big pulse artifact from the TMS pulse. And then when you activate muscle, there's a big decay artifact that if it's a huge amount of muscle you're activating, it can actually bleed into out to 50 milliseconds. If it's a small amount of artifact, it'll come down pretty quickly. So 16 healthy controls. This was our initial TEP with that attenuate protocol. at T4, so the most anterior region, which actually has a lot of muscle artifact. You can see the signal's pretty low. This is what it looks like across targets with the same angle. What you can see is there's a big variation. T6 and T1, the blue traces were the strongest, the most most posterior and most superior. It is very related to the amount of muscle artifact that you can get. So you can quantify that muscle artifact from 0 to 20 milliseconds. The less muscle, the more you can resolve the signal and the larger the signal looks like. And so if you go from the T4, T3 target that we were initially targeting, it's like 1.5 microvolts, and you go to your group best, which was T6, you get that up to 3.5 microvolts. And if you use an individual best, which was for each individual, pick the best target in terms of the highest TEP, you can get that up to 5 microvolts. Basically boosting the signal by threefold. You know, there's a question of what does this signal mean, right? Biologically, we're going different angles and locations, so we're activating different types of neurons. But if we're just looking for the strongest signal, this would be one way to do it. Now, this begs the question, what if we do this in an online manner? Can we personalize more than these 6 locations, right? So this is a study we're running right now, is to determine the necessity and signal-to-noise boost of online optimization. So we developed software to quantify the 0 to 20 millisecond artifact. You can quantify this on a single trial basis, which is different than the early TEP. You often need 100 trials to get an average. So you can imagine a grid search that you can do very easily with the 0 to 20 millisecond muscle artifact proxy. And if that strongly negatively correlates with it, you can go and search for the location and angle with the least amount of artifact, and that should give you the highest signal response in the early TEP. So we've done this in 5 subjects. It's very much in pilot phase. There's a lot of different ways you can do a search, both spatially and angle. There's too many. It's hard to do a brute force grid search here, especially if you're looking at like every 2 degrees and every 1 millimeter. So we're looking at, do we need to optimize angle and location? Can we just take a single brain region and just do an angle optimization and get that boost? Compared to a naive 45-degree baseline. These are some of the different plots we show. Is for each condition, we'll have where the coil is with respect to angle and location. And this is just one plot. I just want you to take from here that this was one subject, one condition where we did an angle optimization, and it resulted in a much stronger early TEP, as we're calling it, between 30 and 50 milliseconds, and it had much less artifacts. This is after artifact removal, so you won't be able to see what the 0 to 20 millisecond response is, but there was an enormous amount of artifact at baseline and very little for the optimized condition. So more to come here. One other thing that comes up, if we can in an online way optimize this as much as possible, is this is a pretty complicated evoked potential. And one question that always comes up in literature is, how do you quantify this, right? There's lots of different ways to quantify this, whether you're looking at quantifying an action potential or something else, it's a curve, right? And I think using a combination of different ways to quantify this signal could be more valuable than just taking the peak within a specific time window at a specific channel, right? We don't really know how that affects the signal. So we're starting to explore these spaces, different time windows around this P30, different ways to pick the peak. Do we look in time or frequency space? How do we define the region of interest? Is it 2 electrodes or 5 electrodes? Is it in sensor space or source space? So there's a lot of different considerations here. And the literature really hasn't done a great job diving into this question. So I hope to have shown you that this early TEP is present intracranially and non-invasively. It reflects cortical excitability. It gets suppressed after a single day and 4 weeks of 10 hertz stimulation. It may be related to clinical outcome, but at least it's related to cortical excitability, local excitability. And we're starting to boost the SNR by suppressing sensory potentials and optimizing experimental and analytic approaches. Just a few more slides here. So if, there's many more studies that need to go into that, but if we assume we can boost the signal to noise to the point where we can get a very reliable signal in the early TEP, then we can start doing the fun stuff. I expressed this to you before, but if we just look at a 10 Hertz treatment pattern, each line here depicts a single stimulation train. I just want you to get that it's complicated, and randomizing 10 Hertz versus 7 Hertz is almost impossible on a large scale. We need to reduce the dimensions, right? We need to ask simple questions like, do single TMS trains elicit acute brain changes that we can quantify, let's say with the early TEP? How long lasting are these brain changes? Is it 1 millisecond or 1 second or 10 seconds? How do these brain changes differ based on different stim patterns? And how do these changes build with repeated stimulation? This idea of metaplasticity is baked into TMS. This idea that a single train, right, each pulse is going to have a cumulative effect. Then you apply 30 trains in a single dose, and then you apply 30 doses in a treatment. The idea is that there are cumulative effects at each time scale that then results in a clinical response, but we have no clue if that's true or not. So we're going to explore this parameter space in this unexplored parameter space. We're going to start with simply looking at different frequencies, amplitudes, and durations of a single train. We're going to quantify that single train, look at the early TEP, and then explore different variations. Once we understand this multidimensional space, both for an individual and across individuals, then we can start thinking about real-time monitoring and closed-loop stimulation, right? You need a library of words to extract, to use, if you're going to adapt treatment. So this is in collaboration with Scott Linderman, who's an assistant professor at Stanford. We've developed a methodology to clean and monitor this early TEP in real time. We're starting to test out closed-loop algorithms in silico and in vivo to demonstrate proof of concept, right? You can think about closed-loop algorithms as Bayesian optimization, reinforcement learning, control systems theory, and within each of those fields, there's a lot of considerations that you need to think about. We're starting to apply closed-loop algorithms to evaluate single-day brain changes on a single dose level, and then eventually we'll build that up to multi-day brain changes and eventually symptoms. So just to conclude here, I had a friend tell me the other day that, not a friend, a colleague / friend, that TMS was a dinosaur. In 5 years from now, TMS wouldn't exist in the clinical space. Tools like focused ultrasound and other treatments are up and coming, and it just won't exist. I want to counter with that I think TMS is just extremely underutilized right now, and we're just touching the surface of what it can do. And I think it can be much more effective. It's also that other methodologies will have the same issues, right? You bring in focused ultrasound, we're going to pick a pattern, it's going to get FDA cleared, and then we won't know how to innovate further. So I think this biomarker approach can be applied for any intervention, right? What we're trying to establish is a way to monitor the changes induced by some treatment. That treatment could be TMS, it could be focused ultrasound, it could be ketamine, it doesn't really matter. But we need a way to assess these treatments. We really want to focus on reliability and validity of this signal. It really is, garbage in, garbage out. I've lived it, I've done it. It's not fun because when you get down the line, you realize that this is not reproducible. And when you're thinking about getting this into patients, there is risk. And so we're going to spend the time on the non-sexy research to try to establish these biomarkers before trying to innovate on these treatments. I hope to have shown you that the combination of online and offline optimization is really critical, that if you have a signal that has tons of noise, there's just no way to clean it up. And so we need to do both. And the combination of invasive and non-invasive methods are really powerful, but also really complex. And we haven't figured out how to combine these. Besides the TMS and intracranial approach, which is simultaneously combine them, how to take results from an epilepsy population and apply that to a different population using different modalities. It's just really tricky. So again, I hope to have shown you that interventional psychiatry is an emerging field with lots of potential, that to personalize and increase its efficacy, we need to find these brain biomarkers, we need to characterize them, and then we need to optimize real-time monitoring for adaptive or closed-loop stimulation. If we can do that, we'll have a treatment with minimal side effects that can be applied to any circuit and any neuropsychiatric disorder. I want to thank everyone involved. It's obviously a group effort. Everyone in my lab, especially want to thank Chris Klein. He's a staff scientist that has been there basically since day one, that oversees most of the operations on the TMS EEG side of things. The participants, collaborators, funding agencies, and thank you for your time. Happy to take questions. See if I can pull up. No. Yeah, happy to. Yeah.
A lot of your work is around biomarkers, like a single pulse, right? It isn't the underlying assumption that you get to effect immediately. So what happens with the TMS treatment after they use the full treatment to actually give the full desired effect, or if it needs time to settle into the new brain state that is the treated state? So what would you, I guess, how would you account for that in this sort of approach?
Yeah, it's a great question. To summarize, if the treatment works by taking a longer period of time to have an effect, the biomarker that we're developing will assess acutely the changes to a single trainer or treatment. So how do you account for that? So there's a couple questions there, a couple things that come up is, and it is a valid concern. You know, to me, is a single dose of TMS the right dimensionality reduction? Are the brain changes that exist after that single dose, does that underlie the therapeutic effect of TMS? You may have an acute brain change that has nothing to do with clinical symptom change. I still think it's worth quantifying that acute brain change and understanding that. So we've thought about what are the smallest timescales needed to quantify this, because I don't want to do a full 30-day treatment of TMS. It may be that it's somewhere in between. It may be that it's 3 sessions of back-to-back theta burst in an hour and a half, and that is the amount of information, amount of plasticity to induce in order to quantify a brain change that directly relates to clinical symptoms. I think your question was a little bit different, is what if it just takes a while to take effect? So we measure typically within at least 30 to 45 minutes of either, if it's a single train, we are applying our single pulses like 500 milliseconds afterwards, 2 seconds afterwards, 5 seconds afterwards. I don't think a single train will last longer than that. The dose, or 3,000 pulses or 600 pulses for theta bursts, we measure basically continuously from second 1 to 45 minutes out. If we see effects that last longer than the 45 minutes, I think that would be fantastic. And then we just need to extend our recording period. If we don't see effects initially, but 2 hours in, those effects occur, which is possible, our approach would be, our approach and other people's approach would be somewhat problematic. Whether you're measuring behavior or electrophysiology, there's some studies that look 24 hours after the last day of treatment. I don't know if there's an example in the animal literature of a type of plasticity that takes more than an hour to take effect, but if you knew of that, I would love to hear it and do some reading on it. But it is a risk for sure.
You mentioned like for the closed loop stuff, like using reinforcement learning approaches to stuff. So my interest is in like deep learning and EEG stuff, and I was wondering if there's any interest or merit in the literature for like trying to use like machine learning / deep learning approaches for the actual biomarker discovery as well. Not just for the closed loop stuff, but even further upstream in the process.
Yeah, it's a great question. Part of the issue with the biomarker discovery is that we just don't have great data, right? We don't have a data set that has hundreds of patients, multiple time points of EEG, and a clinical assessment before and after. And so we just have small sample sizes. So how helpful can machine learning be to help that discovery process? Potentially, and you would think it might be helpful. I'm not sure. The closed loop question is a really interesting one, that you can close the loop at multiple different timescales here. So you can close the loop in the online optimization approach of the single pulse evoked potential. That's a closed loop question, right? Search space of location and angle. How do you reduce that search space quickly? That's just with a single pulse. Then you have trains, right? So what type of stimulation train should be applied? You can't do a brute force search for all the different stimulation parameters. But that's a closed loop question just with a single train. And then there's the metaplastic effect, the single dose effect, which again, you can adapt over sequential trains, right? You can measure every time. So it's complicated, and it's one that we are excited to dive into, starting to think about. Yeah, of course.
I had some questions about your the stuff that you're starting now with the TMS and IEEG. Those patients, are they implanted for epilepsy? Do they have depression? Like, what is that population?
Yeah, so it's the same as the electrical stimulation population. So it's epilepsy population that have medication-resistant epilepsy that are going for surgery. You guys have a big volume of patients here as well. A third of those patients have comorbid major depressive disorder. So depending on how you look at it and think about it, you could either do a subgroup analysis after the fact or just enroll patients with depression. In looking at just the underlying neurophysiology of applying single pulse TMS to the dIPFC, often their seizures don't come from, let's say, the ACC and the dIPFC. I think it was like less than 5% of patients last time we did a chart review. It's usually more mesial temporal epilepsy and other things. So can you study basic neurophysiology that has generalizability to patients with depression or even healthy controls in this population? It's something that always comes up when you study this population, and it is a caveat. You do get great signal to noise and with all the electrodes located right next to the neurons. But that's the question.
I was thinking of it more in terms of like looking for biomarkers, or like something that can be precursors in the EEG work, because if you could also, if you could see like a more direct response in a stimulation target, you can measure like associated neurophysiological changes that happen during TMS.
Yeah, so we're definitely thinking about that. Question is, do you need that clinical response, right? You will get an electrophysiological response to TMS. Can you call that a biomarker if it's not linked to clinical symptoms? So we're starting to think about, can we apply 3 theta burst sessions back to back? This population, you only usually have 1 day where medications are back on right before surgery, where you can apply stimulation, either electrical or TMS. So you don't have that much time, but you could, in, let's say, an hour and a half, apply 3 sessions of theta burst, which may just push the needle a little bit in terms of clinical symptoms, but it's hard to quantify that. So we're hoping a combination of electrophysiology and behavior, that we can start seeing that brain being pushed around in a way that might be valuable. I think someone else.
don't know too much about TMS, but what level of resolution can you get with it in terms of like trying to target a specific area? And to what extent is like depth an issue in terms of like either probing for, you know, different circuits that you're interested in, or, you know, trying to treat a disorder that might be for brain structure.
Yeah, it's a great question. And some of the complications come from the fact that for the disorder itself, we don't know exactly how specific, you know, within the dIPFC, if there was a subregion that we knew was implicated in depression, we could create a coil that targeted that level of specificity. So in general, there's lots of coils out there. They all have a distance and focality trade-off. So there are those that hit a whole swath of cortex that get pretty deep, and then those that hit a relatively specific part of cortex, but only get superficial. These figure-of-8 TMS coils, I think in computational modeling, is thought to maximally activate between 1 to 2 centimeters on the cortical surface. But again, it's still going to hit nearby adjacent regions, just with a little bit less of an electrical current to those regions. So it's not specific, and whether that is helpful for the clinical effect, maybe less helpful for a scientific, you know, rigorous investigation of a specific brain region, but we don't know. So we'd like to get more specific, and we potentially can from a coil hardware point of view, but we don't know if we should. So there are coils out there that aren't used clinically that are much more focal.
It's kind of a follow-up. To what extent do you feel the field is able to assess patients that would benefit from this? Because I feel like to some extent, the field right now doesn't do a good job of getting patients to where they need to go to get like treatment. Like, you have health insurance, and you're like, okay, I have mental health issues. We can look up psychiatrists and psychologists and like where their fields, but that's kind of the patient trying to pick their position. And to the point of that letter that you had was towards the beginning, I feel like patients are just like, I know something's wrong, and I want help. To what extent do we select patients for targeted like TMS therapies? And how well are we able to ascertain which patients would benefit most from get that?
So I wish we did this better, and I hope that's where we're going. Unfortunately, much of the types of people that get into getting TMS treatment is a function of the psychiatrist and how well they are connected to TMS clinics, how good their insurance coverage is there a nearby TMS center. So those dictate patients that fail a couple medications going to TMS or not, more than anything else. Sure. It says, we see the P30 show response to varying TMS parameters. Have you seen similar responses in the power in different spectral bands in areas that are more downstream to the dIPFC? It's a great question, and I'm glad you're seeing some variability with this biomarker. As you know, the ability for this marker to change is necessary in order to develop treatments around it. So, you know, the P30 is pretty low amplitude locally. Downstream, we're starting to look in the parietal cortex, mostly because it's relatively easy to estimate the signal from there. And the frontal parietal network is a core network that we're interested in modulating. It's even lower signal to noise typically. So we do see a P30 in the parietal cortex ipsilaterally, but we haven't seen it vary to different rTMS trains yet. In terms of the spectral bands, I struggle a little bit looking at the event-related spectral perturbation of the early TMS response, I feel that we can extract much of what we need just from the waveform itself. And we know so little about the waveform that to go and do another transform is tricky. And there's also the stimulation artifact, which when you look in frequency space is this impulse function that bleeds pretty far out. So eventually I'd like to look into the frequency space, and it'd be interesting to see what sort of oscillatory activity exists in this P30 or in this early TEP time period, but I haven't done that much. It's a good question. Thank you, guys.